this month's scientific symposium of the PKD RRC. Just to kind of update our administrative portion of this, wanted to make everybody aware of 2 RFAs that are coming out of the center. These are for our pilot and feasibility program, one for early stage investigators and another one separate for the established investigators. The link for that is there. It's up on our website as well. And also be looking for direct emails coming from us on that. We're asking you to save the date, if you will, for our annual PKD symposium. It's virtual again this year, unfortunately, and September 13th and 14th of 2022. So please be looking for more information coming out of the center from that. It'll be up on our website as well. I just want to also make you aware of additional resources that have just become available from the center, which are CRISPR-Cas9 validated guide RNAs for knocking out PKD2. PKD1 will be coming shortly. Also, just wanted to make up the website as a spot for asking your questions about reagents and resources that are available from the center. We have a discussion board up there. Please go up there and use that and start a discussion on a topic that you are struggling with in your research. We're here to try to help with that. Also, if you don't mind, follow us on Twitter. The Twitter account or address is shown there. Again, I want to welcome everybody to this meeting. Stacy, if you want to go ahead and take off that slide. And I'm going to introduce today's speaker, Dr. Untitled-Mukhopadhyay. I want to welcome him and thank him very much for taking the time to visit with us. Untitled is, or soon will be, an associate tenured professor at UT Southwestern, so congratulations on that. You know, if you look back on his career, you see him as one of these pioneers who has kind of been pushing this whole field of cilia-generated signaling. Really tough area to analyze, particularly looking at novel or innovative ways of how proteins get targeted into and out of the cilia. And so today he's going to talk to us about dissecting drivers of ADPKD through the lens of trafficking to the cilia by TULP3. So Untitled, I'm going to turn this over to you. And again, thank you for taking the time to visit with us.
Yeah, thanks, Brad, for that kind invitation. And thanks for having me present this talk. This is a work which is in progress and in collaboration with real pioneers in the field like Carol and Feng. And so the credit goes to all of them and members in my lab. So let me go to people who contributed to the work in the very beginning. Hemant started this, and Sunny, who has been instrumental in taking this forward, Nipunika, who graduated, and Vivek, who is working on the TULP3 project right now. And I am really incredibly thankful to Feng and Carol for their continuous collaboration and insights and all their involvement in this project. So with that, I don't need to introduce you to ADPKD. It's caused mainly by defects in genes in polycystin 1 and 2, and it's imperative that we identify upstream factors. The major problem that I find as a cell biologist to be interesting to me is the defined normal tubule that kidneys have, the diameter, and then the diameter gets increased as they form cysts and there is fluid accumulation. So this is a very interesting problem to me as a cell biologist. Now, I have been working on cilia since my PhD with Dr. Piyali Sengupta in Brandeis, and to me, this is a fascinating compartment. It's very small, size of a few mitochondria. It often projects, but not always, into the external environment. In the kidney tubule, it does project. And it's so small that probably localization of a few molecules in this very small compartment can make really good signaling zones. So you guys all know that there are so many proteins that are now localized to the cilia. It started by papers from Brad and Greg, who showed that polycystins are localized, and then papers from Peter Igarashi and Peter Harris and others, and Stephen Somlo, who all showed that these proteins are now localized to the cilia, including fibrocystin. And others showed that GTPases like Al13B, which are also localized to the cilia, and then GPCRs and adenine cyclases, the list goes on. The main point here is that many of these proteins, if you delete them, they cause cystogenesis, and there is a gradient of severity in the cysts that are from their deletions. Now, this paper has been the most important paper that I have read in this field, and to me it's incredibly interesting, where Somlo's lab showed very nicely that if you delete PKD, there is huge cysts. And then if you delete cilia in the background of PKD genes, the cysts, even in the developmental or in the adult stage, they get smaller, but then they don't completely disappear. And it's similar to loss of cilia alone. So the prevailing model has been that normally the tubule diameter is maintained by a cilia-dependent cyst activator generally being repressed. By then, this cilia-dependent cyst activator gets de-repressed in the absence of polycystins, and this causes severe cystogenesis. And then when you don't have cilia, somehow this cilia-dependent factor still has some leaky function outside the cilia in the cytoplasm. But then it really doesn't explain that how the cysts are even present in the absence of cysts, cilia alone, suggesting that when polycystins are present, this CDCA has to still be de-repressed at this point. So to me, as a geneticist and who do a lot of genetic epistasis, and especially coming from the hedgehog signaling field, it becomes very apparent that probably there are two factors that are counter-regulatory and that are functioning in the cilia in this process. And so one is the activator, which we point to as X, and that is repressed by polycystins in cilia or externally, we don't know. And then there is another factor, a attenuator or negative regulator, that is also present in the cilia, and that when removed doesn't cause that much cysts like from absence of polycystins, but cause less cysts. And so these two factors are present in the cilia, and they function in counter-regulatory modes. Now, this kind of gives us an idea, and this is in collaboration with Dr. Feng Xian, that there is a ciliary component of this CDCA as proposed by the Somlo lab, and that if there is something like this, it's an hypothesis that it has to be localized or trafficked to the cilia, and then its lack should cause no cysts, but then concomitant PC12 loss also should not cause no cyst. Whereas there should be a cilia localized cyst inhibitor or an attenuator, and this inhibitor is also localized or trafficked to the cilia, and then its lack causes small cysts, and concomitant PC12 loss should enhance cystogenesis. Now, these two attenuators and attenuators are kind of similar that you see in the hedgehog pathway, where Brad actually first showed that Glee transcriptional factors are localized to the cilia. But then Catherine Anderson's lab, Jeremy Reiter's lab, Rajat Rohatgi's lab, multiple people have shown that the cilia is fundamentally important in forming both the Glee activator, transcriptional activator, and the Glee repressor in this process. And so thinking in this line, it looks like probably cilia has an incredibly complex role in PKD. And then if we distill the problem, to me, it becomes very complex because there are so many papers in the field where people have shown very nicely that in cystogenesis, there are cellular phenotypes like proliferation, MAP-ARC pathway, cyclic AMP signaling, calcium signaling, mTOR signaling, that are all affected in the cystogenic pathway. But then is cilia really acting very early on in this process? And are there ciliary signals, detectors, effectors, amplifiers that are actually helping in this process that are regulating cystogenesis? Now, that said, it's really difficult to address this because it's very difficult to address what started the process from what is present during the process. It is also very difficult to distinguish the downstream signaling pathways from the upstream ciliary factors. The small size of cilia doesn't make it easy to study with respect to the rest of the cell. And any protein we are studying, it definitely has function outside cilia. How do we actually uncouple ciliary function from extraciliary function in addressing this process? So, but that said, I think Somlo's paper is telling us that, yes, there is definitely ciliary regulators of cystogenesis, if we assume that they're functioning through the cilia and that they're functioning downstream of polycystins, and they could be very important to find out. So my work started as being interested in C. elegans, and this is a movie I took in my graduate years when I was in Dr. Piyali Sengupta's lab. And here you can see the IFTs that are trafficking in the anesthetized head of C. elegans. And this is a real-time movie. And even now when I see it, I get amazed at how prototypical these movements are in the cilia. And these particles are in IFTA and IFTB in two flavors, as shown nicely by Joel Rosenbaum. And these are the particles, complexes I purified when I was in Peter Jackson's lab in Genentech as a postdoc. And the prevailing idea in the field is that IFTB controls anterograde IFT, that if you don't have IFTB particles, cilia are not present or they're really small. Whereas IFTA controls retrograde IFT, that in IFTA knockout, there are cilia present, but there are stumps at the end of the cilia. And so this says that there is a role in retrograde IFT. Now, as I was working in Peter's lab on TALP3, what I noticed was that TALP3 really binds to the IFTA complex very strongly, as much as that currently it's thought to be a part of the IFTA complex. And the binding is actually at the core of the IFT complex, which includes three proteins, IFT 140, 122, and 144. And without these three proteins, TALP3 will not bind to the complex. This binding of IFTA core to TALP3 also brings TALP3 to the cilia. Now, we also found out that without TALP3 binding to the IFTA complex, a lot of proteins are not trafficked to the cilia. This includes GPCRs, including the developmental GPCR, GPR161, which we have figured out to be very important in the hedgehog pathway. And so TALP3 now is thought to be very important in anterograde preciliary trafficking of cargoes into the cilia. It's very important to know that in this process, the IFTA complex is not impacted. So whereas in the IFTA complex, when you are messing with the IFTA complex, the cilia are blunt, like they're short and they are stunted. Whereas in a TALP3 mutant, they are overall okay because the IFTA complex is not being affected. So we, based on the trafficking of multiple proteins, including polycystins and fibrocystin chimeras that we figured out occurs through TALP3, we made this model in 2017 where we proposed that the tubby family protein, which has the tubby domain, is important in binding to the ciliary localization signals, which are diverse. And then the IFTA, the N-terminus of TALP3 binds to the IFTA core. And this is how this tripartite complex is trafficked into the cilia. Once inside the cilia, because there is less 4,5-PIP2 in the membrane, the tubby domain dislodges from the membrane, and then the cargoes are retained in the cilia, whereas the TALP3 IFTA complex starts to traffic on its own. Jeremy Reiter's lab and other labs has also nicely showed that if you change the PIP2 composition of the membrane, proteins like TALP3, IFTA, and the cargoes of TALP3 starts to get accumulated in the cilia, also favoring this kind of a dislodgement in the cilia. Now, I would say that this idea of IFTA trafficking things into the cilia is still not thought to be thought to be very important in the field, because This is a line from a review last week, where this kind of movement is still thought to be exception to the rule of bidirectional IFT transport. The reason I'm telling you this is that it's so clear that IFTA does retrograde transport in the cilia that we really don't think that IFTA through TALP3 is really fundamentally important in trafficking things into the cilia. And so we have a lot of work to do in this line if we are correct. So we really wanted to understand that if TALP3 can help us in this manner, in understanding the role of the ciliary component of CDCA or the attenuator CLCI. And so we did a knockout of TALP3 in collecting ducts in developmental stages, and we saw huge cysts at P24. We were very excited because we just knew that TALP3 regulates polycystins in trafficking to the cilia. But then we realized, and we also saw that the cilia in this process were not affected as expected, that the cilia length or the amount of cilia in this model was not affected. So that was very good to us, that the cilia are intact and that we can now study factors that are important in PKD. However, as I went to the PKD meeting and I talked to others, and they immediately said to me that these cysts were really small compared to other PKD cysts. So then we really staged them in development, and we saw that the cysts in the TALP3 conditional knockout was similar to that from loss of cilia, as nicely shown by Peter Igarisi's lab years before. And that if we now have a double deletion with PKD knockout, the animals die a day before. So it's definitely causing more severity in end-stage renal disease, but the cyst size are not changing a lot. So at this point, we collaborated with Dr. Feng Xian, and Feng's lab has this beautiful model where they have a hypomorphic PKD1, which actually is blocked in autoproteolytic cleavage in the GPS site. And these PC1, they don't go to the cilia, the V format. And then when we made a knockout of TALP3 in the background of these PKD VV mice, what we see was that there was an enhancement of the phenotype in the cystogenesis. So this was also kind of interesting because a paper from Peter Harris's lab recently came out. And in this paper, Peter Harris's lab and many others in the field, they showed that IFT core, IFT140, is a very important gene in the PKD spectrum, and that the cystogenesis in these patients is mild. They have limited kidney insufficiency, and that this could be one of the common groups after PKD 1 and 2 in causing PKD spectrum disease. So this was also very interesting to us because our developmental models, which in TALP3 was enhancing the PKD phenotype and the role of IFTA complex in cystogenesis, similar to TALP3, was very interesting. There's also a paper from Greg, where Greg Pazur nicely showed that loss of IFT140 causes cystogenesis, and in a way similar to TALP3, but in those cases, the cilia are almost completely very stunted. So if you take this with the idea of CLCI I put before, you can make a model where you can think that maybe TALP3 is regulating a CLCI to the cilia. That becomes more apparent in the PKD1 VV model. And that's why if you have the cilia intact, the cysts in PKD mice are not reduced, but rather it's enhanced when you have both these factors impacted. However, we don't know what these factors are or how many factors could be present. A parallel paper came from Carroll's lab, and Carroll's paper beautifully showed that in the adult kidney model that he used, and where he knocked out TALP3 throughout the tubules, he saw that the cysts in the PKD knockout were really attenuated at like 4.5 to 5 month stage. Very late in adult stage, TALP3 knockout did cause cystogenesis, but at this stage, this was acting as a real inhibitor of cystogenesis, the loss of TALP3. And another paper from Pamela Tran also came out on IFT subunit in IFT 139. And in this paper, the Tran lab nicely showed that when they knock out this peripheral IFTA subunit throughout the tubule, they see that there is a decrease in cystogenesis in this adult cystogenesis model. Now, again, going back to to Greg's paper, where they showed that cilia was really gone in the IFT 140 knockout. In the IFT 139 loss kidney, the cilia are normal length, as you see from their data. So it's not that the cilia are gone, but the cilia are present. Of course, there will be defects in retrograde transport in the cilia, but it is saying that this subunit is impacting in PKD1-2 knockout phenotype. So again, taking these data together, Carol's data and Pamela's data, it kind of suggests that again, the TALP3 with the IFTA complex core could be regulating a factor, the ciliary component of the CDCA into the cilia. The good thing about the TALP3 models is that the factor in the cilia is being affected. The factor outside, the same factor outside the cilia remains intact. So this kind of gives a handle of what is happening inside the cilia. So what cargoes of TALP3 could function as CCDA or CLCI? That's the million-dollar question, and that's very interesting. So very early on, we did knockouts of GPR161, which is a very important negative regulator of HOX signaling. Conditional knockouts of GPR161 does not cause cystic kidney disease. We know that this is present in kidneys at these stages, also from recent papers where they have really nicely showed that GPR161 is one of the GPCRs that is present in the kidneys at almost all stages in the tubules. So we started looking into other factors, and one of the proteins, Al13B, which is a small GTPase and used in the field as a ciliary marker, was being affected in this, and Carol saw the same in his models. And in this figure, what you see is that the cilia is marking in green. The cilia is actually, this is not in PP5E. The cilia is marking in green in the middle. It's not in PP5E. It's acetylated tubulin, and you see that the cilia are present in the middle panels, but then Al13B is completely lost in the cysts. But then you can see them in the adjacent tubules where it is still present in the TALP3 knockout. So we are doing a TALP3 knockout only in the conditional ducts, in the collecting ducts. And you can see that Al13B is being affected in the collecting ducts alone. So Al13B has been studied by multiple labs, including the Wittinghofer lab, Tamara Kasperi's lab, and Rick Kahn's lab. And the model they have proposed is that this functions as a GEF in converting Al3 into a GTP form. And then this Al3 GTP acts on effectors for carriers for farnesylated and myristoylated proteins in releasing them into the cilia. So we started looking into farnesylated and myristoylated proteins that have been previously proposed to be present in cilia and important in cystogenesis, like in PP5E, a farnesylated protein, and NPHP3 and cystin-1, which are myristoylated proteins. We also looked into other farnesylated proteins like LKB1, which are not Al3 regulated. And we first looked into kidney cell lines, and we saw that NPP5E was really affected in the TALP3 knockout. NPHP3 and cystin enrichment was affected in the ciliated cell lines, whereas LKB1 was not affected. So we looked into these factors in the kidney, and it's very encouraging because these factors, the lipidated palmitoylated Al13B protein and the farnesylated and myristoylated proteins, they all have mild developmental fibrocystic phenotypes. And that suggests that this could be important in regulation of ciliary signaling at this stage. So when we looked into in PP5E, what we see is that the cilia is marked in red here in the collecting ducts. You see that at this stage at P5, when the cysts are starting to form, that in PP5E, which was present in the cilia and controls, is missing in the TALP3 knockout. Now, if we look into NPHP3, this is at a much later stage, like P24, when the cysts have started and formed pretty well. At this stage, you can see that acetylated tubulin cilia are marked in the middle, and you can see NPHP3, which is very much present just distal to the transition zone in the inversion compartment. And then what you see is that in the TALP3 knockout, you don't see them localized to the cilia. So saying that NPHP3 is being depleted from the cilia, but slowly. So if we draw the kinetics, what we see is that LKB1 is not lost at all from the collecting ducts in these kidneys, whereas IL-13B, which is in green, is lost very beginning, like even before the cysts start forming. And in PP5E is slow to start, but then it drops by P5 when the cilia are forming. And NPHP3 is very slow to decrease over time. We could not look into other markers like cysteine-1 because we were not able to look using antibodies for these. So bottom line, it looks like IL-13B and or a regulated factor like in PP5E downstream of IL-13B could be very important as the attenuator in this process, which functions from the cilia. Very encouragingly, we also got this IFT140 knockout cell line from Greg, and it's a MEF. And in this knockout MEFs, we see that as expected, only 20% cilia remaining. But in those 20% cilia, we see that both IL-13B and the GPCR, GPR161 are missing almost with only 20% remaining. So this is suggesting that these cargoes are also being affected by the IFTA core in the remaining stumpy cilia. So this says that TAL3 IFTA core could be regulating these cargoes, especially IL-13B dependent proteins in the CLCI activity. And so we are really looking into other these cargo candidates like in PP5E, IL-13B, in using genetic epistasis in the PKD1 conditional and in the PKD1VV conditional backgrounds to see if they cause enhancement in the developmental stages and or if they cause repression in the later stages. So if they do, then they could be good candidates for ciliary CDCA or CLCI activity. So, but you know, there could be multiple factors that are trafficked, and any factor that we are knocking down could also have extraciliary functions. And if you are trying to find out multiple factors traffic to the cilia, it's a very difficult problem to address. And in this case, we have to have other ways by which we can target the ciliary component of CDCA. And so the one way as cell biologists, we got very interested in this process is that, is there a logic behind all these diverse ciliary localization sequences that so many people have discovered to be important in ciliary localization in GPCRs and in other proteins? Like, you know, Karp-Mikitin showed long time back that there are these sequences in GPCRs that are really important for trafficking. We showed that GPR161 has a CLS. Greg Pazur's lab showed that there is a nice fibrocystin CLS that on its own is necessary and sufficient for trafficking. And then what is the CLS for IL-13B? Now, this was a very new protein that we were working on. And so Vivek started looking into how this protein is trafficked by TAL3. So what he found was that when he did direct binding experiments with in vitro translated MYC IL-13B here and GST TAL3, he saw that only the C terminus of the GST TAL3 was binding to MYC IL-13B, whereas the N terminus was not binding, saying that the TAL3 C terminus stubby domain is important for MYC IL-13B binding. He also purified IL-13B from insect cells, and then he did the binding reaction between GFP IL-13B and GST TAL3, and it binds nicely to GST TAL3, but it doesn't bind to the control GSTB. It's saying that this is a direct interaction between these two proteins. Now, before, when we were working with GPCRs, polycystins, fibrocystin chimeras, we could not do direct binding like this because these are all transmembrane proteins, and we really did not have a handle on this binding, but we were very satisfied to see this binding directly between these two proteins. So how is this binding happening? So this is a structure of TAL3 of IL-13B from alpha fold. And what you see is that there is an N-terminal amphipathic helix, and this is the GTPase fold. And very strikingly, when we started making deletions for TAL3 binding, we saw that when we deleted like when we had the C terminus of IL-13B, it was not binding, saying that the N terminus of this protein was important in binding to TAL3. But then Vivek did a very interesting experiment. He took only the N-terminus amphipathic helix and then fused it with the C-terminus. And in this case, he saw that the binding was restored, saying that the binding is probably being regulated by the N-terminus of this protein, which is an amphipathic helix. This amphipathic helix is a lipid clamp. There is a palmitoylation motif here, and this can regulate the GTPase activity of this protein. So most of the time, people in the field, they actually don't even include this when they are doing their GTPase assays because this messes with the protein. So we went and started looking into this helix in very detail. And basically what we started doing was deleting parts of this helix. And we saw that the initial amino acids before palmitylation and the second batch of amino acids after palmitylation are both important in binding, which we are showing here in the delta 1-2 mutant. You see that the binding doesn't happen with the, as you see in the full length. Of course, if you delete the first 19 amino acids from the full length, the binding does not happen again. And then when we looked into the helix, this has a distribution of charges and hydrophobic amino acids on the other side. So this is the moment of the helix of this protein. And so we started making deletions so that we removed all the charges, and this is the 11A mutant, whereas this still keeps the palmitylation motif intact. And in this case, we still see that now it doesn't bind to the protein at all, to TAL3 at all. Whereas if we start doing mutations in the hydrophobic amino acids here, so that they're all charged residues, they still bind. So it suggests that charges on this side of the protein are probably interacting with the TAL3. This is very interesting because it starts help us develop models by which these diverse ciliary sequences are structurally related in binding to TAL3. And so an idea is that TAL3 has this amphipathic helix, and that charges on this side of the helix are important in binding. For GPR161, we also know that there are charges on this side of the protein, and we have actually now deleted these two Ks only, and we see that this is not trafficked to the cilia, suggesting that this is the region that is actually interacting with TAL3. For fibrocystin, and this is known to be in the alpha fold structure to be actually a part of the TM5, which extends outside as a helix. For fibrocystin also, the CLS is very close to the extended transmembrane helix. And of course, this structure is so complex, it's not yet in alpha fold. So we don't know, but we predict that this would also be part of an extended helix, that the lysines here are really important in binding to TAL3. So this gives an idea that structurally, the helix is probably important, and it has to be a charged helix, which is important in binding to TAL3. Now, correspondingly, are there one site, two sites, multiple sites in the TAL3 TABI domain that are important in binding? So we don't know that. So we started looking into sites that are modified in TAL3 by these ciliary localization signals. And the way we did it was by mass spec, where we took these different GPCR CLSs, like for GPR161, and we collaborated with Steve Gigi's lab, and we figured out what are the lysines in TAL3 that are modified. Now, this is not a direct binding assay, but a proximity assay, where the lysines close by this CLS are modified. And we found some promising hits. And very interestingly, these all fall on one surface predominantly in the protein. And that was very interesting to us. And if you look into this now, so the TABI domain is actually a beta barrel, and the beta barrel has these 12 strands of beta strands, which are around this helix, and the mutations that we are getting are on one side of this beta barrel. And so that's very interesting. The other lysine that we got is actually on the side of the protein. So one of these lysines was K389, and I was in this meeting with Carol, and Carol showed to us that the K407 mutant, which is identical to K39, also had a phenotype. This is before he published the data. So we were incredibly lucky because he shared this data with us. And that said that this mutation, the mouse TAL3K407I mutation, was also causing cystogenesis. However, the mechanism by which it was doing was not known. So by two methods, by this forward genetic screen from Carroll and by our biotrilation screen, we found the residues in that side of TAL3 that was important in TAL3 function. So we started looking into different cargoes that are in proximity to TAL3. And one of the cargoes we now know is AL13B. So in this experiment, we are showing that AL13B is in proximity to TAL3 using a proximity biotylation assay. When we have a TAL3 mutated in the K389 site, we see that AL13B is not in proximity to this now. And the same is for another cargo like the fibrocystin CLS found by Pazur. In this case, we show that the fibrocystin CLS is losing proximity if we have the K389 site in TAL3 mutated. So again, saying that both the cargoes are in proximity to the same site in TAL3. This is very satisfying to us because it's saying that there could be one site that is regulating these cargoes being trafficked. So then we looked into all these different cargoes that are trafficked by TAL3, and that includes AL13B, INPP5E, GPR161, fibrocystin CLS, and also other GPCRs. And the way Vivek did this experiment was to take a TAL3 knockout and then express the wild type or the K389 mutant, and also the I and the A mutant we have done in different flavors. And what we see is that in every case, he sees that although TAL3 is expressed, and this K389I goes to the cilia, the cargoes are not trafficked. So this is saying that this site is really important in TAL3 for trafficking. Now he has looked into so many different functions of TAL3 like IFTA binding, PIP2 binding. These are all unaffected in the K389I mutant. And only cargo binding is affected, as I showed you. And so that suggests that this could be a very important site for trafficking of TAL3 cargoes. And we have been looking into many other mutations in this beta barrel region and the H12 helix in order to figure out what is the logic behind how TAL3 is interfacing with all these different cargoes. But this also gives us an idea that this could be a way we could drug TAL3 in the context of ADPKD. The idea being that there could be small molecules, peptides, or small molecules that are binding to this region and inhibiting trafficking of TAL3 cargoes. Now, peptides are incredibly difficult to introduce into cells, so we really need to have small molecules. But if there is one site and if multiple cargoes are being affected by this one site, the CCDC, a component of this PKD, can be affected by this kind of a strategy. And so in a very preliminary experiment, what Sunny did was that she took the SNAP lines we have. Now the SNAP lines are lines where we can see trafficking of really newly translated protein into the cilia. And so these are not the end state ciliary cargoes, but this is trafficking only ciliary proteins that are being trafficked over 6 hours. And she adds these peptides, which are myristoylated. And so we can see them move. We expect that they will go into the cells. And then she has both SNAP AL13B GFP and SNAP GPR1 GFP lines. And when she introduces these peptides using this approach, and these are incredibly messy, very little peptide is probably going into the cell. But very interestingly, she is seeing that AL13B GFP entry into the cilia is being affected by a fibrocystin peptide. And similarly, a GPR161 trafficking into cilia is also being affected by a fibrocystin peptides. Now, these are no way close to where we can take these as inhibitors of TAL3 trafficking, because if you look into the full GFP component, you will barely see any difference. But it gives an idea that this area, this region of TAL3 can be targeted by different, in different contextual formats by these peptides or by small molecules. And that says that maybe this could be a druggable target. So what I try to show you today is that TAL3 traffics multiple cystogenic lipidated cargoes to the cilia, which are good candidates for CLCI. And that is a good thing and a bad thing, because there could be multiple CLCIs and it would be difficult for us to figure it out. But I also showed you that in an alternative approach that there are structural similarities in the ciliary targeting regions for diverse cargoes, and those are probably helixes with charges that are helping in interacting with really defined regions or a single pocket in TAL3, which is really exciting from a structural perspective and in figuring out and probably drugging TAL3 as a candidate for ADPKD. So with that, I would like to thank everyone. I really am not in the PKD field. I don't know much. I'm learning from these meetings. And so it's a privilege and an honor for me to actually present our work here. Thank you.
Okay, that was wonderful. Thank you very much for a wonderful talk. I'm going to open this up for questions. And I agree with Lisa's comments about the wonderful slides telling a beautiful story. So thank you for doing that. It is open for questions. And while the first ones are coming in, oh, sorry, go ahead, Lisa. I see you want to say something.
So thanks, Brad. So I sent Brad a note, Seket, to say what beautiful slides and what a magnificent story. The question I have, you know, I was intrigued by the NPHP3 and the slow progression of cysts as a myristylated protein. Cystin's also a myristylated protein, and it's very rapid progression. And so the interplay between those two pathways, can you speculate about why you see such a difference in terms of the pace of cyst development?
Yeah, so, okay, so let me go back to this slide here. Here, can you see my screen?
Yep.
Yes, so, okay, so cystin and NPHP3, so I mean, you know everything about cystin, and we have really tried to work on cystin based on your papers. But NPHP3, as much as we know, is very much localized to the inversing compartment, which is a proximal zone of the cilia. And it's integrated there by other factors. So there is like a sink that keeps it there. And those proteins together help in maintaining it. So whenever we do like knockdowns, like in our knockout cells, what we see, let me pull up the slide. So here. So in these cells, when we are taking IMCD3 cells, and this is a TAL3 knockout, and I did not show these slides, but when we do a knockout of TAL3 here, we see that cystin is still present, but very less amount. And if you find out like it's still present, so because, you know, whatever TAL3 brings in, there's also a sink by which it is retained there in the inversing compartment. So I speculate that this is the reason why some of these myristylated proteins are being removed from the cilia so slowly in our model. And regarding whether these are CLCI, I don't know. I mean, there could be so many factors, MPP5E, cystin, NPHP3 that together are working in a combinatorial mode in functioning in the ciliary membrane. And there could be like, maybe based on what we see to begin with, NPP5E is really important, but in later stages, maybe cystin and NPHP3 has a role. And we really don't know how to put this together at this point, but I'm just suggesting that there could be a combinatorial role of these proteins in this process. Yeah.
Thank you. Other questions? Can I add a question on the structural motif?
Yes.
You know, there's always been this question of, you know, the cilia targeting sequences are different for every protein you look at, right? So based on this kind of structural motif that you're putting together, are you able to predict what membrane proteins might be ciliary targeted versus ones that are not?
No, not yet. That's
Okay.
the thing. So, okay, so there is no like, this is the thing now. This is a second level. So we can't predict it from the sequence. What we can predict now is that this K and this K is important in this helix for trafficking. So for all the extended ICL3s, we have to actually do the hydrophobic moment of those and really try to figure out if there is a distance between the charge residues, which are important in trafficking to the cilia. And it's nothing to do with the baseline sequence that is going to tell you the answer. So the secondary helix will go tell you the answer. We haven't really done this. Like there is, we have a friend here, a collaborator who again found another GPCR that is going to the cilia. Like over the weekend, he just emailed me and I can't see the sequence and tell what are the residues there. But if I do the hydrophobic moment on these, I can probably start putting charges which are important in that helix that is important. I think that's the logic.
Okay. Dr. Sun, I know you had your hand up and then.
Yeah.
A beautiful talk. I read your paper on top 3, but I forgot whether polycystin trafficking is affected.
Yeah. Polycystin.
Question. But,
Yeah. So polycystin trafficking is affected in our knockout cells, as you see in this slide, and fibrocystin chimeras are also affected, and then when we look into like proximity assays, again, which we also published, we see that for polycystin 2, this proximity is very much dependent on the TALP3 binding to the membrane. And so this is a TALP3 KR, which is PIP2 deficient in binding. And so you see that the binding reduces a lot when you don't have PIP2 binding. So the idea is that TALP3 is regulating these cargoes through PIP2 binding. And polycystins also have other targets, just like IL-13B, ciliary targets like RVXP motif, as you well know, in IL-13B, which is also important. The interaction between the CLS, we don't have a CLS for PC2 yet. It's incredibly difficult. We couldn't figure it out. So the CLS, at least in IL-13B and the RVXP motif, they interact very strangely. So we are still figuring out how it does. So I am suspecting that there are similar functions of how TALP3 is regulating PC2 trafficking in the cilia. Yeah.
So a follow-up question. It's really interesting that the AIL13B is completely missing from cilia, because previously we thought the reason for the mutant to develop cyst is because they lost the cilia. What happens if you make a double mutant between AIL13B and TALP3?
And TALP3, yeah, we are doing those experiments. So we are actually doing genetic epistasis in developmental modes with PKD knockout. Sorry, it did not come out clearly. That we are doing it with the PKD conditional knockout in developmental mode and with PKD-VV with Dr. Feng Xian, and we are doing it in the adult modes. We are seeing very interesting results with in PP5E in the developmental mode and with IL-13B in the adult mode, but I did not put those because they are work in progress. And your point is very important because multiple papers, your papers and other papers from Tamara's lab, they all showed that the cilia are lost in the IL-13B conditional knockout. And in this case, at least in the TALP3 knockout, we see that the cilia are present and IL-13B is lost. Now, in these models, when we are doing the genetic epistasis, at least at the stages we are looking, we want to be very sure that the cilia is not lost. And the reason why the cilia is lost in the IL-13B knockout, but not in the TALP3 knockout, is probably related with TALP3 having some redundancy with TABI and other factors that are important, at least in the developmental stages. So I think these are all will factor in when we are doing the genetic epistasis. That's why I did not put those data in, but yes, we are doing all those experiments. Yeah.
Yeah, that's great. Thank you.
Dr. Zhao? You are muted.
We can't hear you.
Yep, you're muted.
Okay. Thank you for a very beautiful talk, Saket.
Yeah, thank you.
I probably missed it. Zhao Xia asked a little of the questions I have, but I was wondering about the polycystin 1 and also the polycystin 1 trafficking, whether it was affected by the TALP3, and also whether you looked into the ciliary trafficking motif we identified in polycystin 1.
Yeah, polycystin 1 is also affected so both polycystin 1 and polycystin 2 are affected by TALP3. And we haven't looked into your motif. I know we haven't. So the problem is that whenever we try to take a CLS and we try to contextualize in a chimeric form, it's not always works. So I'm sure Hemant, who was doing these experiments, again took your construct and he may have tried, but in many cases we are not able to, like for example, the somatostatin receptor CLS, we cannot do it in a chimeric form when we take it. So in all cases, they are not always working in our hands. And so we have not been able to, and polycystins always, polycystin 1 and 2 are interacting. And so at least we know that polycystin is affected by TALP3. We don't know either the CLS for TALP3, for PC2, and for polycystin 1. We would predict that it would be in one of the intraciliary domains and and I mean, I know your sequence, but we haven't really looked into it carefully. We should do that, yeah.
So in this figure you are showing us right now, are you showing polycystin 1, or am I just missing it, or?
No, we are not showing polycystin 1.
Oh, okay.
Figure, yeah.
Okay, so you didn't show it. It's not I missed it. Okay.
No, not here in the figure, yeah.
Okay.
Any other questions?
Or if no one is asking more questions, I actually would like to explore a little bit further. When you use, you said chimeric constructs, I was wondering what kind of chimeric constructs do you use?
Yeah.
How much do you include? And,
Yeah, exactly.
It matters, and we have shown that.
Exactly. Yeah, I completely agree. And so it's something to do with the CLS being an extendable TM helix. So for example, here we do a CD8, which is the fibrocystin CLS, and the CD8 TM domain is extending with the fibrocystin CLS. And in this format, it works as a cargo, but other formats, I don't know how much it will work. So we try to keep it to this format, but in many cases, we can't get, so as much as I think that for TAL3, you need a helix to form for it to actually interact with the cargo, and it has to be close to the membrane. Although you are pumping a lot of protein by your expression methods. So I think that would be the ideal way to do the experiment, and to have it very close to the membrane in an extendable TM format. Yeah. It's a hit and miss. It doesn't work in many cases.
So in summary, so you mentioned that you have identified a number of cargoes, ciliary cargoes for TALP3, and which might mediate both positive and negative signals. Mm-hmm. So I was wondering, based on your analysis and your data, are you suggesting that we just need to look in this group of molecules?
Yes.
I think you have included all the candidates. Now we can narrow down instead of looking at a bunch. And now you have helped us to narrow down these signals are actually within these less than 10 molecules or whatsoever. I don't know how many actually there are, but maybe you can remind us.
Yes, I mean, that's the idea. The problem, however, is that there could be so that's the idea. Now, there are a lot of farnesylated and myristoylated proteins that are trafficked to the cilia. And so we have been looking the well-known ones here in this list. And of course, INPP5E is a real promising candidate because we already see that it is lost from the cilia very early during cystogenesis. So this is a genetic epistasis experiment that we are doing. The problem with the genetic epistasis experiment is that we are losing whole INPP5E and we are not losing INPP5E from the cilia. So that again is not same as in TAL3 knockout where we are losing INPP5E from the cilia. So whatever data we get will not confirm that. It will suggest that INPP5E could be a CLCI, but it will not say that it's a, and so because we don't know these, it really makes the list smaller, but it's also difficult because we can't do a lot of combinatorial genetic epistasis experiments. We can do one at a time. Yeah.
All right, second, I want to thank you for a wonderful talk. The last part I wanted to bring up was, we are a consortium that is meant to build resources for the community. And so I'm going to reverse this and ask if there is anything that the PKD consortium could do that would help advance your research or areas that you think we should focus on to help push this story forward.
Yes, yes. I mean, we are terrible in 3D cystasis. I think anything in the context of human kidney cell lines from proximal tubules or collecting ducts that could be done for PKD genes and our candidate CLCI components of CDCAs would be a big help. And especially in the context of introducing CLS variants of these candidates in these cell lines. So I think that, and that would also help us in any small molecules that we probably get in our assays.
Okay. So I'd encourage you, if you think more about this, that there's specific things you want us to generate. So, the subcommittees will take this back and they'll discuss how they can push this forward. But if there's anything specific that you think that would really help in this field, please don't hesitate to reach out to us and help us generate these models.
Yeah.
And resources you need. All right, I want to thank you again for a wonderful talk. I want to thank everybody for attending. The next meeting is a month from now, and be looking out for the emails for the next science talk. I appreciate everybody participating and wonderful discussion. So thank you, everybody. Saket, thank you again.
Good. Thank you.
Thank you.